Well, first of all, I'd like to thank Andrea for this invitation, and it's a great opportunity to share our progress with the colleagues. So my name is Dapeng Zhang, and I'm based in Beltsville. My project works on genetic diversity analysis of cacao, coffee, and other tropical crops. But today, my presentation is about authentication of coffee varieties using sleep markers. How do I move to the next
Just
one?
let me know when you want to move.
Could you move to the next one? Yeah, thank you. Next, please. Mhm. It's moving or it's not?
Did it not move?
Did you say the?
There
Yeah.
you go.
All right, thank you. Sorry. So one of the major problems we know in coffee production and coffee research is genetic integrity. That's a lack of genetic integrity. So the mislabeling is a serious problem in germplasm collection, in breeding programs, and in farmers' nurseries. And based on the published literature and reports, the mislabeling rate is alarmingly high. For example, in the Cantier Arabica collection, you have about 39 to 91% of mislabeling, depends on which variety, which accession. In the Robusta coffee, you have about 18.6% in the example we show in the Gala Robusta collection. And based on WCR's report in the seed lots from Central America, we have about 36% of the seed lot that have high rate of mislabeling, which means they have a mislabeling rate more than 50%. And the one who has a good genetic conformity is only 26% in the seed lot in Central America. The main reason for this one is because coffee, they don't have a different from maize, wheat, soybean, and other major field crops. They don't have a good formal seed system, right? And also is because of their biological feature of this unwanted pollen contamination and unwanted segregation, which I will talk about later. So to tackle this problem, we need to have a simple tool, which is should be cost effective, suitable for large scale downstream application. So we started by developing a low density panel, we call the sleep genotyping technology, using the publicly available sequences. So in this case, we use the sleep markers developed by the French group. Then we screened about 1,000 different primers for Arabica and for Robusta. And then we selected a core panel for Arabica coffee genotyping and genotype identification. And here is a truncated form for the Arabica low density panel. And here is a truncated list for the Robusta coffee genotyping. Both has been used in large scale for germplasm identification, breeding stock verification, and seeds lot authentication. So far, in terms of germplasm collection, with the collaboration of Tracy, Chinji, Minnie, Luz, Ricardo, the colleagues in ARS system, and outside the system, we genotyped the HARC collection, University of Puerto Rico collection, ECAF collection, Cantier International collection, and various farmers nurseries with collaboration with World Coffee Organization. We also genotyped a bunch of Robusta collection in Craig, Ivory Coast, and Ghana. The technology, it's easy to be applied because we basically outsource most of the genotyping to service company. All you need is to have this sample kits filled with the dry leaf discs, and then you send them to the service genotyping service center, and then they generate data and send it back to you. So in this way, any lab in developing countries, even they don't have the molecular facility or setup or skill, they can benefit from this technology. So this is an example of the slip fingerprints we generated for Arabica coffee. The top one is called Anna Coffee 14. The bottom one is IH90, both the Catimore type of coffee. What a surprise to us when we saw the fingerprints was that we noticed that there's a high rate of intra-varietal segregation. We thought as a self-in species, it should have a highly homogenized homozygosity along all the fingerprints, but actually they are not. So this actually is one of the fundamental problem for the high rate of mislabeling in Arabica coffee, which we will show later to how to tackle this problem. So I will give a couple of case studies to show the effectiveness and the usefulness of this technology. So this is for the Cantier gene bank. We genotyped about 2,400 trees representing about 1,800 Arabica accessions, which by far is the largest Arabica collection in the world. And so we removed the duplicate and the redundancy, and there's about 1,700 unique genotypes, out of which 484 represent the whole Ethiopia wild population. And so we selected a core set for USDA-ARS introduction, right? And the core set represents three stratifications. The first one is wild Ethiopia, which we consider as a first priority. We probably will include all of them in, we should include all of them in the USDA-ARS collection. The second stratification is the varieties introduced from Africa, particularly East Africa. And the third stratification was the one from the other countries, such as Brazil, Costa Rica, and everywhere. Now, the problem probably now it's the core set is there. We know which genotype we should introduce, but the challenge is and what form should we introduce clonal material or should we introduce seeds families? If we introduce seeds families, the problem is then you will need a large number of seeds to represent each mother tree. And that will be a huge burden for gene bank management in Hawaii or Puerto Rico in the future, right? So we will discuss this, continue to discuss this and refine the correct strategy, which should be cost effective and accurate. My second case study, the example I would like to give is about this Limani and the Fronton variety from Puerto Rico. The Limani is a, how do you call, Sachmo variety. That's a Timo hybrid typed with Villa Sachi. And the Fronton is a variety of Catimore. It's a Timo hybrid. crossed with Catula. These are the two dominant variety in Puerto Rico, and they have a good flavor, they adapt well, and a good disease resistance. But however, because of the hurricanes that destroy 80–90% of the whole coffee production and nursery in Puerto Rico, so those varieties are gone. It's even difficult to find the authentic source of material to recover them, right? So that's a major problem. With the collaboration of Luz and Ricardo and other Puerto Rican colleagues and World Coffee Research as well. So we collect about 200 farmers' tree from different farms. And also we collect about 200 trees from the Puerto Rico University collection. So these two graphs show the first 50 accessions. The top one is Fronton, the bottom one is Limani. So the light purple color, actually it's Limani, and the yellow bar represent the Fronton. So as you can see, lots of Fronton were labeled as Limani, and lots of Limani were labeled as Fronton. So there's a high rate of mixture. More than that, you can see there's lots of mislabeling the hybrids. They are neither Fronton or they are neither Limani, right? So based on this result, we select the so-called pure line from each varieties. And then we compare with the reference Sachmo and the reference Cantimore, such as Costa Rica 95, Marsellesa, and different reference varieties. And we confirm that these two varieties, Puerto Rico variety, have their unique genetic identity, and they can be recovered. So that we can go back to the original tree and select the most representative pure line start propagation. So this is the second example. Again, this is in collaboration with Mini. We show that this tool can be used to rapidly identify, confirm the F2 segregation, the mapping population, which Mini and Chi are working on this one. And this one, I believe this sample came from Adriana, but I think I received this from Tracy. So we can identify their putative parentage from each tree. I think I received about 300 or so in total from Tracy, where Tracy said those trees have resistant phenotype in farmers' field, but they want to know what are the genotype, what are the varieties they belong to, things like that, right? So the genotype is still going on because our system broke down. So the whole process slowed down, lagged behind. I want to apologize for Tracy and Adriana, but this is based on 230 Arabica tree I received from Tracy. Those are the inferred parentage of those 203 trees, which has putative rust resistance. So this is not to say that in this case, we have 36% of parentage coming from Cantimore and Sachmo. Then the second large group is from Tupi and Yellow Bourbon. Then the third group is from Obata. So here I'm not saying this is the Obata variety or this is a Tupi variety. What I'm saying is there are the parentage, the pedigree came from those varieties. So which indicate there are different source of resistance in those 230 trees. Of course, lots of things can be followed up on those varieties, on those trees. For example, we need to find out, are they still segregating, or are they stable, purified already? And what are their bean quality and other things? So this indicated that in addition to the breeder's material, there might be some other farmer selections that can be we can work on and find out if a potential source of new varieties. So as we can see from the previous slides, the source of resistance so far, everything pretty much came from Timo hybrids. It's a very single, narrow scope of resistance. How long those resistance can last, even in those resistant genotype, we don't know. What I'm trying to point out here is that the Arabica diversity is just a very tiny group of diversity in the whole Coffea gene pool. We have many other source of resistance, which we may take advantage and use them in breeding program, right? So here shows the tiny Arabica center of origin from Ethiopia and Southern Sudan and Bowman Plateau, but then we have a huge gene pool for Califora and for Liberica and for many other species. So finally, I would like to just show one example. That's Coffea stellophila. This is a rare wild species originated from West Africa, from Sierra Leone, from Ivory Coast and Guinea. Right? What's good about this species? It has a good flavor comparable to Arabica, but it's adaptable to low elevation and has good heat resistance and drought tolerance. So they are more robust than Arabica. We genotyped 3 populations from Sierra Leone, just want to understand how much diversity are there. And we show that there in Sierra Leone alone, at least there are 3 populations. However, in the gene bank, the national gene bank, they only have 1 population. The other 2 populations are completely missing. So with this result, we show that we can fill the diversity gap by systematically collecting the wild populations before they disappear, right? And we also show that the population is really in danger because in the top, we show that the species has a very low effective population size. And on the bottom, we show that a major bottleneck happened almost like 25,000 years ago. That's during the last ice age, the last glacial maximum period, which probably narrowed down the habitat of this species and reduce the effective population size from 4,000 to 1,000. So that sort of will provide evidence to show why people say this species is endangered. Yeah, so urgent rescue measure need to be taken before those species disappear. So this species actually can be either directly used, be crossed with each other, and also they can be crossed with Liberica, they can be crossed with Califora as well. So the potential can be used as a new source of disease resistance and adaptability and quality as well, right? So with this, I will just finish my my talk. Yeah, thank you.